guys, and welcome back to Biologue. Hope you are having a great day and a great week. Now, as you can see from the title, this week's video is going to be an IGCSE past paper video based on medicinal and recreational drugs. So we're going to look at some questions on these topics. Now, unfortunately, these will be my last few IGCSE past paper videos, but I will still have topic wise videos coming up. So this will be either IGCSE or IB or VCE topics, and we'll go in a synchronized order of that syllabus. So make sure you leave your topic suggestions in the comments section below, and I will make sure to add them to my list. And without further ado, let's get into this video. The fungus Trichophyton violacea reproduces asexually by releasing spores. A single spore was placed in the center of a petri dish containing an agar medium with starch and protein. Now, figure 1.1 shows the fungus that grew from the spore. All right. So, state the name given to the body of the fungus that grows from a single spore. All right. So, how this works is that you have these single strands called hyphae. All right. Remember. Now, this is something I typically get very confused with, so make sure you guys remember this. Hypha is plural, hyphae is singular. So, hyphae is the single thread or the single strand that, when compiled together, it's called hypha. So, hypha is plural. Now, each strand is called hyphae, and this combines to give you mycelium, which is like the entire body of the hyphae. All right. So there is mycelium. So the body of the fungus that grows from a single spore is called mycelium. Alright, this is called mycelium. Now the next question, let's see. The thin threads that make up the body of the fungus. Exactly, this is what we talked about right now, which is the hyphae. So your answer should be hyphae, but because it's asked you for thin threads, remember this is where wording of the question really makes a difference. Alright, if it asks for thin threads, it means plural because the S is the threads, right? Many. So it's asking for the plural version of hyphae. So you have to write hypha. Alright, typically they will give you marks if you still write hyphae, but it's always better and safer to make sure you write the correct one. Alright, so the next question. Describe how a fungus such as T. violaceum obtains nutrients from the agar medium. So, how this works is that the hyphae that we just talked about, it releases these digestive enzymes. Alright, hyphae releases or secretes digestive enzymes. Alright, and what do digestive enzymes do? These digestive enzymes, like, you have to give examples of what kind of enzymes there are, like amylase, protease, etc. These are the types of enzymes that could be there. So you need to write secretes digestive enzymes like amylase, and what does amylase do? Which breaks down starch to maltose, as well as proteases, proteases that break down proteins to amino acid, amino acids. All right, it breaks down proteins to amino acids. All right, and these amino acids, or these the maltose, any products such as the amino acids or the maltose. Alright, this is how you're supposed to write it in a very full length form. Or the maltose is absorbed back into the fungus, as nutrients into the fungus for nutrition purposes. Alright, for nutrition purposes. How does it get absorbed back? By diffusion. Alright, it can either be diffusion or it can be active transport as well, because you don't really know the concentration of each of the substances in and out of the fungal cells, or in and out of the fungus. So you can say diffusion or active transport depending on the concentration, right? Or depending on the concentration difference, depending on concentration gradient or concentration difference. All right. Moving to the next one, Streptomyces, a soil bacterium, is a major source of antibiotics that are produced by pharmaceutical companies. An antibiotic sensitivity test can be carried out to help doctors decide which antibiotic should be used to treat a specific disease like gonorrhea. Now, gonorrhea bacteria isolated from a person are grown on an agar medium, a ring with 8 different antibiotics. This is very important. This is a key statement here. With 8 different antibiotics is placed on the agar medium and left for 24 hours at 35 degrees Celsius. Figure 1.2 shows the growth of the bacteria on the agar medium after 24 hours. So when you look at the figure, one main thing that you see is that there is bacterial growth, and in some regions it's clear, right? It's like white in color, it's clear. So that wherever it is white in color or clear, it indicates that there is no bacterial growth in these regions. But wherever it is darker shaded, that is where there is bacterial growth. So let us now look at the question. Suggest why there is a clear area around some of the antibiotics. So there is a clear area around some of the antibiotics because now most of the bacteria would have been killed, right? Where the antibiotic is present. That is why there is no bacterial growth. So it would have stopped the reproduction of the bacteria. So the antibiotics, alright, would have stopped the reproduction of the bacteria, or would have killed the bacteria. Now, as there is a ring surrounding the antibiotic, would have killed the bacteria. Alright, so as there is a ring surrounding here, alright, we can actually say that the antibiotic must have diffused through this agar medium. Alright, so as the antibiotic must have diffused through the agar medium, must have diffused through the agar medium, as shown in the diagram, or shown in the figure, Alright, let's look at the next one. So explain why antibiotics 1 and 5 would not be chosen to treat the gonorrhea infection. Look at 1 and 5 right now. In 1 and 5, there is absolutely no clear region around the ring, right? Around the antibiotic ring. And this means that there is maximum bacterial growth, but no, lack of bacterial growth. So none of the bacteria has been killed in that case. Or basically that. So in antibiotics 1 and 5, none of the bacteria has been killed, so the bacteria has grown around the disc because of the black color region, right, the darker region. So the bacteria has grown, or bacteria has not been killed by antibiotics 1 and 5, hence has grown around the disc. Antibiotics 1 and 5, hence they have grown around the disc. It has grown around the disc, Alright, so the bacteria must have become resistant to the antibiotics 1 and 5. Must have become resistant to the antibiotics, hence 1 and 5, and hence antibiotics 1 and 5 were not chosen. Hence antibiotics 1 and 5 were not chosen. Alright, this is your answer. People who take antibiotics should always be told to complete the treatment rather than stop taking the antibiotics when they feel better. Suggest why people are given this advice. Now, you have to talk about the role of antibiotics. What exactly do antibiotics do? Alright, antibiotics are these substances or these types of drugs that help to kill bacteria, right? So why are people given the advice that antibiotics need to be completed? The whole course of antibiotics should be completed so that all of the bacteria gets killed. This is done, so you need to write it like this. This is done so that all of the bacteria gets killed all of the bacteria gets killed, because sometimes even after the person becomes well, there might be still some bacteria present in that body or in the person, and so this might cause higher chances of replication for that bacteria. And so because it is able to continuously grow, now it might actually become resistant to the antibiotics. So that's what you're going to write. This is done so that all of the bacteria gets killed, because sometimes even when a person becomes well, even when the person becomes well or feels well, when the person feels well, the bacteria may still be present. The bacteria may still be present, and if it continuously replicates, it can become resistant to the antibiotic. It can become resistant to the antibiotic. Alright, so this is your answer. This type of question can be easily molded into a paper 6 question or an ATP question. So make sure you guys pay attention to this question, because if I was the examiner, I would have easily molded this type of paper 4 question. So typically, I think examiners, they take paper 4 questions, especially the concepts or ideas that have a little bit of experimentation knowledge required, and they just mold it, change the wording, and they keep it in paper 6. So this is something that can definitely happen. So I will make sure to practice these types of questions as well. So you guys need to make sure that you pay attention to this part of the video. When bacteria are spread onto agar in a petri dish, they form colonies. Each colony forms from one bacterium. Figure 4.1 shows an investigation into antibiotic resistance in a species of bacterium that causes disease. Now you have these diagrams given. There's no antibiotic added to the agar in A, and there are colonies of bacteria which are formed. Transfer of bacteria culture into dish A, and here is the culture. One second. Yeah, here is the culture of bacteria in a liquid medium, and that is transferred to dish B. And in dish B, the antibiotic is added to the agar. This is then transferred from dish B into another liquid medium. And this liquid medium, again, the bacteria is taken, the culture is taken and transferred to dish C for 24 hours. And antibiotic added to agar. Okay, and here in this one, it is taken and then it is exposed to X-rays. This is quite interesting. So what exactly would happen is what they're actually prompting you to think about. Now this is then transferred to dish D, and antibiotic is added to the agar. Okay, so you have 4 dishes, 1, 2, A, B, C and D. That's 3 and 4. Okay, moving on to the first question. So the first question asks you, what is meant by the term antibiotic? And here you have to write the definition. So because it's only 2 marks, you don't have to elaborate so much, but you have to be very careful with your words, as in you need to be very concise, because it's only a 2-mark question. So the examiners won't be searching around for keywords, and they won't be wasting time on that. So you need to be really concise in definitions. Alright, so an antibiotic is a drug or a substance. Alright, an antibiotic is a drug or a substance that kills bacteria, Alright, an antibiotic is a substance that kills bacteria, and now you can think of other things. You can say it kills bacteria, or it stops the reproduction or growth of bacteria. Stops the reproduction, or in bracket you can write the growth of bacteria. It stops the reproduction or growth of bacteria. So those are your 2 points. First being that it's a drug or it's a substance, and second that it kills or it stops the reproduction of bacteria. Alright, moving on to the next question. Explain why only a few bacteria grew in dish B compared to dish A. So it's a 1-mark question. Be very simple. What is it asking? Only few bacteria grew in dish B compared to dish A. Look at dish B and dish A. See the difference. So now this is basically because in dish B, we know in both cases, see, look at this. In dish A, no antibiotic was added, meaning there was nothing that could be used to kill the bacteria. Whereas in dish B, antibiotic was added, so there was something that could be used to kill the bacteria. So in this case, in dish B, you need to write that since there was antibiotic present, most of the bacteria must have been killed by the antibiotic. Most of the bacteria must have been killed, been killed by the antibiotic. Most of the bacteria must have been killed by the antibiotic since it was present in dish B. Alright, since it was present in dish B. Since it was present in dish B. Alright, this is how you're gonna write your answer. Alright, moving to the next question. More bacteria grew in C than in B. So now you have to look at dish C and dish B. Where is C? Here. Alright, this is dish C. Now in this case, antibiotic was added to the agar, and in dish B also an antibiotic was added. So it's not the presence of an antibiotic. Here also the absence of an antibiotic would not matter because antibiotic is present in both. So what is it that makes C different from B? Alright, in C there were much more bacteria because in B, there was fewer bacteria which became resistant to the antibiotic in B. So because of which most of the bacteria has gotten killed in B. That's what you should see here, right? Most of the bacteria has been killed, and as a result of this we can infer that so in this case only some bacteria must have become resistant. Only some bacteria would have become resistant to the antibiotic, So this is your answer. Alright, this is how you're supposed to think. Look at what is given in the diagram. In the diagram you can see there's very few bacteria present, right? Most of the bacteria has been killed. So write that down. Most of the bacteria has been killed. So what can you infer from this? When we say most of the bacteria has been killed, it means that the antibiotic has been effective in killing the bacteria. So only some of the bacteria, very few quantities or very few amount of the bacteria would have become resistant to the antibiotic. Alright, so that is your answer. So more bacteria grew in C than in B because you had most of the bacteria became basically died or got killed in B. So most of the bacteria got killed in B, hence less or few bacteria became, or only very little, or basically few is a better word, only few bacteria became resistant. Actually instead of few I would say only some bacteria became resistant to the antibiotic in B, compared to C, which was more. Alright, moving to the next question. Figure 4.1 shows the effect of an antibiotic on a species of disease-causing bacterium. Suggest why antibiotics should not be used very often. This is very important. Now this type of question asks you, why is it important that you should not be consuming antibiotics without a proper prescription from the doctor? And why is it bad that you actually, if for instance if you take it too many times more than it's required, why is that bad, right? So it is bad because now you're giving repeated exposure to the antibiotic for the bacteria. So now the bacteria gets repeatedly exposed to the antibiotic, but not all the bacteria gets killed. That's really important. Alright, but not all the bacteria gets killed. Alright, and because not all the bacteria gets killed, you have some of the bacteria becoming resistant. Some bacteria survive and hence become resistant to the antibiotic. Survive and reproduce, hence becoming resistant to the antibiotic. Some bacteria survive and reproduce, hence they become resistant to the antibiotic. Okay, so that is your next answer. Alright, moving on to the next question. Explain the possible effect of X-rays on bacteria. So remember, what is X-ray? X-ray is a mutagen. A mutagen is a type of chemical that causes mutations, or basically, could cause mutations. It's a sort of substance or chemical that could cause mutations, and this can be radiation, which is like X-rays, or it could be other substances that actually can be cancer causing or can be mutation causing. Alright, so what you need to write here is that X-rays are a type of mutagen. X-rays can be a type of mutagen. Alright, hence they can cause mutations in the bacteria. And now when you say mutations in the bacteria, you have to explain what mutations are, which are a change in the base sequence of the DNA, a change in the base sequence of the DNA in bacteria, or of the DNA itself, okay, a base sequence of the DNA, okay, which are a change in the base sequence of the DNA. Hence, this mutation in the bacteria can lead to antibiotic resistance. In the bacteria can lead to antibiotic resistance. This is why mutations are actually really harmful or really bad, right? It can lead to antibiotic resistance, maybe causing a new strain, maybe causing a resistant strain of bacteria to be produced. A resistant strain of bacteria to be produced. Alright, now let's look at the next question. So it says, state 2 ways in which the structure of a bacterium differs from the structure of a virus. Alright, so remember that, bacterium, you can say that bacterium has a cell wall, whereas a virus does not have a cell wall. Typically, bacteria is a prokaryote, right? And remember that all prokaryotes, most of the prokaryotes have a cell wall of a very complex structure. It's called a complex cell wall, which is made of a mixture of protein and carbohydrate. And this cell wall mixture is called peptidoglycan. Alright, so this is not something you need to know, but it's always good to know. So I would say that you should know that the prokaryote cell wall, or the cell wall of a bacteria, typically is made of peptidoglycan, which is a mixture of protein and carbohydrate. So you can say that bacterium differs in structure by the cell wall, which is a complex cell wall made of peptidoglycan, but you don't really need to know the peptidoglycan part. Just say that it's made of a different type of material, so the cell wall is different. And you can also say structure of a bacterium differs from the virus in the sense that you can say flagellum, right? The bacteria has a flagellum, which is the tail-like structure that helps the bacteria in movement or locomotion. Alright, moving to the next question. Human immunodeficiency virus, or HIV, which is a sexually transmitted disease. It infects the cells of the immune system. Now describe the effects of HIV on the immune system. This question is really important, and I'll tell you why. It's because this type of question, it's taken from the last part of your chapter. So when you go through the chapter, you will see that this HIV is in the last part of the textbook, and typically people tend to ignore that part. So always examiners will try to take the part that is usually ignored, so that they really see who has gone through the chapters well enough that they understood the whole chapter. Alright, not just like the main portions of the chapter. So that's something you need to really pay attention to, the specific details and things that go unnoticed in the chapter as well. So let's go to the question. So describe the effects of HIV on the immune system. So remember that the HIV, it sort of affects which cells in the body. It affects the cells of the immune system, right? So which cells? The HIV affects the lymphocytes of the immune system. Alright, it affects the lymphocytes, and the lymphocytes, remember what they do is that they produce the antibiotics, they release the antibiotics. So because of this, fewer antibiotics fewer antibodies. Sorry about that. Lymphocytes, what is the function of lymphocytes? Lymphocytes secrete antibodies, all right, which help in gaining immunity against a pathogen. So lymphocytes, they affect the lymphocytes, so fewer antibodies are secreted or released or basically produced, also any word should be fine, because of which infected cells, of which, when a pathogen enters the immune system, when a pathogen invades a cell, the infected cells are not killed. All right, infected cells are not killed or not destroyed. So as a result, what happens? Since they are not killed or destroyed, now the phagocytes, all right, and phagocytes, why? Because remember that the lymphocytes and the phagocytes, they go hand in hand, right? Lymphocytes release antibodies which attach to the antigen of the pathogen. And so you have, let's say you have a pathogen here and you have an antigen. This is the pathogen, or the bacteria or the virus, basically the foreign material that comes into your body. It has a substance here called the antigen, and then you have the cell, which is your lymphocyte, and it releases antibodies, all right. And these antibodies are complementary. Remember, the shape of this area is an active site. It's sort of like an enzyme substrate complex where it needs to bind and have a complementary shape, right? So the shape here is complementary to the shape of the antigen in the pathogen. So it binds and then it neutralizes the pathogen or kills the pathogen. Either it does this directly, or it asks for the help of the phagocyte, right? So the phagocyte is the one that engulfs the pathogen into its vacuole and then digests it or breaks it down. All right, so when you look at it in that sense, now because the infected cells are not destroyed, the phagocytes cannot engulf them effectively, engulf and break them down effectively, all right. Hence they are not as effective as usual. They're not as effective as usual, due to which what happens? All right, due to which if a disease is contracted, if the person contracts a disease, the recovery time is much slower, as in it takes a lot more time for the recovery to occur. It contracts a disease, a lot more time is taken for the recovery to occur. Lot more time is taken for the recovery to occur. That will be it for today's video, guys. I hope you liked it. If you did, do make sure to like, share and subscribe to Biologue for more such biology, exam revision and productivity content. And I will see you guys in my next video. Have a great day.